Central Molecular Research Laboratory at Biochemistry Department is one of its own kind in whole Uttarakhand. It is the single molecular laboratory in Deharadoon offering broad range of PCR-based tests. Walking starts with the dress changing area. After wearing the dress, proper air shower should be taken. Area 1. The physically separated area required for the preparation of molecular biology grade reagents. For example, for the PCR based tests, we prepare the reagents in this area. The area is utilized for liquids preparation, nucleus-free water utilization for reagents preparation, etc. Preparation of the reagent should be stored after preparing in 0 degree centigrade for further processing. Now, in area 2, after finishing the work in area 1, we will enter into the area 2. This is also called as sample preparation area or DNA-RNA isolation area. For PCR-based diagnosis, DNA or RNA from the clinical samples can be strictly isolated in this unit only. Area 3 or amplification area is the unit where the target gene DNA sequences of the pathogen from the clinical samples, DNA RNA from the clinical samples can be processed further for PCR, which is called as amplification process, which is being utilized for routine diagnosis of Mycobacterium tuberculosis, commonly called as Koch PCR or TB PCR, HLA-B27, DNA profiling, RFLP, RAPD, AFLP, SSR markers, and many more. The real-time PCR machine from Quigen, RotorGeneQ, is one of the best real-time PCR machine which is approved for molecular diagnosis of swine flu, H1N1, herpes simplex virus, 1 and 2 detection, hepatitis C virus, genotyping, which mainly includes HCV genotypes 1, 2, 3, 4, 5 and 6. It also does cytomegalovirus quantification, qualitative detection, chronic myeloid leukemia, molecular characterization by studying BCR-ABL genes, human papillomavirus genotyping of oncogenic types is done by this RotorGeneQ machine. Then we also is having Roche Cobas Techman 48 real-time PCR machine, which is IVD, FDA and CE approved instrument for the molecular characterization of various pathogens, which includes hepatitis C virus RNA quantification, hepatitis B virus DNA quantification, HIV type 1 RNA quantification, CMV cytomegalovirus DNA quantification, etc. The results obtained from this machine is highly accurate, reliable and rapid, as Roche is one of the approved instrument worldwide, especially utilized for viral load monitoring in patients. We are having the area 4, which is also called as post-amplification area or area for result interpretation for molecular based diagnosis. This area of the central molecular lab is used for agarose gel electrophoresis, SDS page, reverse blotting, gel casting and gel documentation for the PCR based results. The most important asset of the central molecular lab and advanced molecular diagnostic center, which is having equipped with biosafety level 3 facilities. This lab is a single approved center for swine flu H1N1 testing in whole Uttarakhand. This swine flu testing center is approved by National Center for Disease Control, New Delhi Government of India and Ministry of Health and Family Welfare New Delhi, Government of India also. Before entering into the BSL-3 lab, a person must wear proper personal protective equipment with N95 mask, shielding goggles and gloves. Entering into the BSL-3 lab for H1N1 sample processing includes putting the air handling unit on, where the person will first of all move into airlock unit 1 with the negative pressure of minus 10 Pascal, then moving into the airlock 2 with the increase in the negative pressure of minus 20 Pascal, followed by the main working area, which is having the maximum negative pressure of minus 30 to minus 40 Pascals. The biosafety laboratory is well equipped with biosafety cabinet B3. It is an approved instrument that is required for sample processing for suspected cases of swine flu H1N1, which includes nasal swab and throat swab. The instrument is utilized for inactivation and pre-processing of the nasal swab and throat swab for further swine flu testing, which includes RNA isolation from the clinical samples, etc. The area is well equipped with cooling centrifuge, vortexer, spin win, etc., where these instruments are utilized for the sample processing, inactivation and disposing of the waste material of BSL-3 lab. The unidirectional workflow must be maintained, and after finishing the work, exit from the lab will be from the different direction, following again the gradients of the negative pressures. Samples of BSL-3 lab, nucleic acids, mainly DNA and RNA, must be stored inside the laboratory itself, which is having minus 70 degrees centigrade for its long-term storage and further processing, as any kind of the infectious material should not be taken and stored in the general lab area of BSL-3 lab. At the end, the personal protective equipment which a person was wearing should be properly discarded in an autoclave bag and should be autoclaved in a double door horizontal autoclave which is mounted in the BSL-3 lab, and after autoclaving it should be completely burnt out. Now the person will take the proper shower and wear clean room dress and come out of the lab for further processing of H1N1 samples and result interpretations.
जिंदगी है कोई तो हो राज़दार।